G'day, good evening, thanks for listening. This is an horticultural gardening show, focuses on plant science, soil science and microbiology, and the information provided is for educational and scientific purposes. It's fact-checked and the information can be relied on, and the papers or any websites and things that are discussed will be put in the description below. And if you're watching this back, you might want to fast forward until you see the screen change. That's when the show will start. You'll see some slides happening, and I'll give it a few minutes, another 70 seconds odd, for cool folk to arrive, like Infinite Wonders. Hey, says Infinite Wonders, and Small Tubes. Cough, 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 always says Small Tubes. Hello, hello. Nice to see you. And after this show today too, there's a study session on crop breeding and plant biotechnology. I've got an exam coming up in 3 hours' time on that subject. It says the basics of crop breeding, but holy mackerel, it goes into fair income detail. It's just the basics because it's got a molecular stuff, which is at the end, but the stuff at the start's pretty relevant. Back crossing, test crosses, and dominance, and all sorts of things. Ploidy, it's great. So that's going to be the study session after this show today, which is pretty much going to follow up straight afterwards. I nearly didn't want to do this show today because I didn't want to get my brain into another mode, because this exam's going to be hard. It's very detailed, knowing complete dominance, over dominance, all its sort of different applications that you can get for breeding. I hope I pass it. But, all right, 2 minutes is up. Talking about today's show, which is it's the first on the principles of seed technology. Share screen. Principles of seed technology. This one. So it's a shortish course. It's not usually 12-week one, which is what the normal ones are. It's an 8-week course, and the first 4 weeks just is so detailed. It's really good. The last 4 weeks are in particular crops. So today, how about we go through the principles of seed breeding. Principles of seed technology. These are the slides for it. Sharing screen. Do I have to wait any longer? Stop sharing screen. I think I'm set up now, which is good. To stage. Oh yeah, I can change the screen to that one. Make it bigger. Yes, that's better. And thank you to Blinky for becoming a member. If you want to become a member, you're welcome to support it below. It's all appreciated because I don't get anything from doing all this, putting this effort in. So I appreciate those who can contribute. If you don't contribute, that's still fine. Just share the stream and that'll make me happy as well. If you're poor, like me, back to it. Let's start the seed. Clicking too many buttons here. There's too much stuff on sharing screen now. Good. Try to start it twice. So it doesn't really describe the course at the start. Well, there's eight weeks, 40 modules. Agronomy, physiology, maintenance, breeding, biotechnology, plant protection, processing, and storage. Actually, that's pretty much what's going to be covered in the seed technology. Yeah, because there's a lot of. That's it. This is. Can you go any brighter? No. It's pretty dark, isn't it? Sorry about that. But that's a seed. A ripened ovule or a fertilized mature ovule containing embryo which has developed after fertilization. So that's all it's got. It's bits on it. The micropyle. Actually, this isn't the best for you to picture of the micropyle. The helium, the big bit that you always see on the outside of it, it's detached. Then it goes inside of what there is everything inside. The radicle, first little white bit that you see that comes out of the seed, that's called the radicle. Isn't that rad? And then it forms into the root after it goes down. Anyway, let's get a bit of diagram. What's this say? The dry or wet dispersal unit, fruit, with matured ovule developed after fertilization. So here's different plant types. Might be even a maple, that one there. See? Those wings. That's another cool thing this we'll talk about, seed dispersal, transport. How it gets around, and you can see by the maple, it's got wings on it. It's cool as. A propagule responsible for maintaining the intrinsic or genetic qualities of variety or hybrid. Okay, that's showing us that probably you can propagate plants by doing that with bamboo. Ginger, I think you can do it the same way like that. You just cut it off and it grows. You plant it horizontal and it grows upwards like that. Bamboo. And also, if you want to get into how you can make money off this sort of stuff, look online and see how much it is for a bamboo plant. Well, if you're in Australia, I bought one because I couldn't believe the price. The cheapest I could get was, I think, $70 for a little small thing. Like massive amounts. In my shop, the major retailer here sells them for $100 to $200 for the smallish. They're not big. I thought, holy mackerel, there's got to be some money in that. So yeah, there is. All you got to do is grab them, plant it like this, let it come up, advertise it. This mostly goes to the States. So Craigslist, bamboo. Keep your variety and your little sticker thing that you get with it when you purchase it, so you can put that up as your selling thing. And this goes on to say, an embryo, which is a living organism embedded in supporting or the food storage tissue of the protective coat. So inside seeds, they're live. That's why it's hard to germinate ones that go dormant, because they've gone dormant too much and you can't pull them out of dormancy. There is techniques, which will be discussed later on. It's pretty cool. What's that? Seed. It doesn't even say the tester, which is the seed coat. It doesn't go into much detail. And it should say the starch stored for the cotyledons for the food. That's the starchy bit, which is most of the seed. And the amylase, we'll get into that. Any propagated material, miniature plants, dormant plants, maybe that's showing the seed size and colour. You get all different types of, especially the corn in particular. This, I've got some great red stuff at the moment, which I'm going to try next year and see if I can grow some red corn. I've only had a few kernels that come out that were red, so I'm keeping them. Maybe some kidney beans up there. And it goes on to see, we'll go through it later on, the seed size is characterized. What's this say? Link between two generations, carrier of improved or modified genetics. Well, they're synthetic seeds. They've been started. What do they call encapsulated seeds as well? They're sodium, calcium carbonate, and sodium high. I've done a video on how to make them. Sodium. Sodium. No, I'm not going to guess. Starts with G, I'm pretty sure. But yeah, that's what that slide says. Encapsulated, they're pretty cool. They store for a while. They sponge down. They're very tough. It's another way of starting plants. Generative part of a plant that develops into a new plant, a mature embryo embedded in synthetic nutritive gel. You make up those two compounds, and then you add a solution of nutritions to one of the compounds, and then you put into the other, and then it drips and it forms. When it drops into it, it forms this ball. It's pretty cool. Just instantly, it just falls in from the reaction, then it drops it in. So the stuff that you've dropped in, which would be little bit of this green material that you want to start your plant with, and the matrix and stuff like that, and it drops in and it forms, whoop, goes straight and encapsulated into this instantly. And you pick these up in your hands, then they're hard to squash. Seed structure. So it's getting about to the bits on the plant that reproduce, like the female bits on the stigma at the top, then the style, and then the male pollen. This is a dioecious plant that produces both of them, both sex parts on one plant. So, yeah, I'm not going to elaborate on how it, well, the pollen grain goes into the actually, I'll just continue. The style, the female bits, and inside there, down integument, the embryo sac, the egg cell, and the ovary. That's where it should say the micropyle is down here, the bottom bit now. Then the mother eggs in the middle that form the triploids. How a real carpel looks. Okay. That's pretty cool. Here's a cartoon version of it, of the carpel. You can see the male and the female bits on it. It's got it, the style and stigma, petal, anther, and the filament. And receptacle, okay, very good. Some more cartoon, good to go in a bit more detail. Here we go, a bit more detail. Fertilization of the egg. It's where it happens, where the pollen tube, the pollen grain lands on top, travels down, puts out a, this might go into greater detail, puts out a calcium signal that follows the nuclei down around, goes in through the micropyle and fertilizes the egg. If it goes a little bit past the egg, that's where it can hit the mother cells in the middle, which have a 2N chromosome count, not a 1N on it. That's where you'll get the 1N from the pollen grain with the 2N, making it 3N, which is your triploid. Or it just forms a normal diploid because it's got 1N coming around here with the 1N that's already in there. That gets you your diploid fertilization. Here's a better view of it. So the pollen grain comes down, it's the stigma, puts out the sperm with the nuclei there, travels all the way around, right around here, up through the micropyle, to say micropyle, no, and then hits the eggs inside of the ovary. And that's when one can travel a little bit further and go in here, and one sperm cell fertilizes the central cell, which adds which is 2N to start with the 1N coming in, which equals the 3N. So that's the triploid, where this is a normal diploid fertilization. That's one way of forming ploidy. Let's see if there's any, what did the screen change? Oh, no way. Can't see anything. Oh, gee whiz. Thank you for that. Oh, gee. Can you see now? You can see here. I have to start again. Should I start the whole stream again? I'll start again. Fast forward to, so you can see this now. Yeah, you can see this. All right, we'll have to go back. There's tree structure, agronomy, physiology, maintenance, breeding, biotechnology, plant protection, processing, and storage. That's all the things going to be in this course. A ripened ovule is, or a fertilized matured ovule containing embryo which has developed after fertilization. You can see some bits on the outside, bits on the inside here. That's where I was saying before about the radicle. It's that little white tip that you first see that comes out for the start of germination, which is pretty cool. And this is the dry or wet dispersal unit, fruit with matured ovule. I'd like to thank Infinite Wonders too for saying that. Yeah, really appreciate that. I'm going to stop now. It works again. Can see, thanks. Excellent. That was terrible. Hey, there's Lene. Hello, Lene. Hope you're doing well. Yes, and can see, thank you. Good. Thanks for picking me up there. It's only at the start. Oh, that's all right. I did mention at the start of the stream for the rewatchers to skim through till they see the slides change. And that's what I was saying about the maple leaves. Those ones talk about seed transport later on. I don't need any birds under the windowsill. It's right there. An embryo, which is living organism. That was the last one when I was explaining about the bamboo root. Ginger grows similar ways, and you can make money if you want it off bamboo. Just go get a plant, cut a bit off, or if you've got it growing in your region, you have to find out what type it is. So you'd want to be taking photos and getting the exact type it is. Maybe get when it fruits, when it's got flowers. Those types are the easiest ways to get descriptions of them. And then you can sell it. So all you do is cut it off like that, put it in the ground, and it grows vertical. That's how they propagate bamboo. Well, one of the easiest ways to... Go away, bird. Sorry about that. Woolly wagtail. Yeah, and I was saying about the prices of the bamboo too. The reason why why you could look into it is because in Australia, $70 for a 8-inch plant, miniature. I bought one of them. And I thought this is something you could make money off down the track. Just let it grow for a few seasons, get big, take cuttings. All of a sudden, you've got 10 plants up, which is worth, well, put a zero on that. That's good effort money just for listing it. And this goes mostly to the States. So like a Craigslist example, or in Australia, it might be Gumtree, you could put it on. And before you know it, you've paid yourself to buy another plant, a more expensive one. So this slide says, an embryo, which is living organism embedded in the supporting agent for the food tissue and the protective coat. So there's the tester, which is the seed coat on the outside, and the stored food is the starchy bits, which the seed is probably at least 70% starch. Depends on the seed. They got a lot. Some seed has oils in it too, like I think the peanut plants has 50% you can get out of it. It's quite a high amount. It all depends. This one says about the different sizes, like the corn, the blue corn. I presume it is, or it looks like corn. So I got blue corn, dark and light blue, and I got red and white. So I'm going to try and grow some red stuff next year. This is about the encapsulated synthetic seeds. I've made a video on it. If you are interested, you can go and see how I did it and the 2 ingredients you got to buy to do it. That's it. On a dripper, because you need a dripper to drip one into the other to form a little blobs. It's pretty interesting. It's the genetic part of a plant that develops into a new plant. A mature embryo embedded into the synthetic nutritive gel. So you'll have your sodium, gelatinate, calcium carbonate. Mix them all together. You don't mix them together. You drop one into the other, and that's how you get the droplets. The seed structure. So this is the dioecious plant, which has 2 bits on it. The monoecious only has 1 bit, 1 reproductive part. So you get your males or your females with the dioecious species. This is the 2. And that's right. I'll go, it just shows the bits of the carpel, the flower bit. And then it's a cartoon depiction of it, which shows pretty much the same thing, which is just in cartoons, not as, it's all right. And now we get into the more detailed bits, fertilization of the egg. So that's when that's right, this one up here was better for describing it. Pollen grain lands on the stigma on the top. It puts out a cell sperm nuclei, which is a calcium signal at the front of it. That travels all the way right round up through the micropyle in the ovary and hits the and looking for the cells inside of it. So there's the egg nuclei cells down here, which are 1N, and the central cells, or the mother cells, they're 2N. So if you get your pollen tube, which is 1N, pollen grain, one set of chromosomes, and it comes in, it pollinates the first ones, you'll get 2N, which is a diploid. Which is your diploid, pollinates those ones. And if it pollinates the middle mother cells, the egg cells in the center, that's when it's 2N plus 1N, which is 3. So it's a triploid. So it's one way of fertilization forming. I'll stop again. I'm so glad I stopped there. Just to ask, doing all right, thanks, says Lene. Hope you're doing good as well. Busy, thanks, because I've got this exam coming up after this live stream. There's another live stream too in blooming 40 minutes from me. So I'm only going to go through this first week of these slides. I'll do next week tomorrow because I've got another live stream. All right, sharing screen. I'm sure I am. No, I'm not. Now sharing screen up top. Yep, good. So what's this say? Oh, this goes into name the different cells that are inside of it. So the egg cells are down here, synergids, and the antipods up the other end. This is the micropyle end where the nuclei, the sperm nuclei fertilizers first comes in. This, the embryo sac, that's what that's breaking down. Parthenocarpy. Parthenocarpy, what's that? Well, it's an ovary that may develop into fruit without fertilization. Examples of that are banana, papaya, orange, grape, apple, and pineapple. You can also do parthenocarpy with gibberellic acid, which is one of the phytohormones. Here we go into all over the shop, but it's showing the tester this time, which is a seed coat. Cotyledons, they're the first two little leaves that develop that to support the rest start of the plant. So theoretically, there's enough starch inside of this tissue to break down with amylase. And then the amylase extracts or produces amylase, which is the enzyme to break down starch, which produces the ATP through the sugars that it goes through the process, and then it gets its energy, and that's enough to make the cotyledons germinate and go green. So once they go green, then they can start and produce the rest of the hypocotyl, the stem, and that's enough energy. So theoretically, you don't really need any energy at the start, and especially more so if it's an organic seed. If you source organic seeds, that's how the germination will happen. And if you source seeds that have been used to being fed by their NPKs, you might get different processes where it might fade quicker because it might be used to getting bottle fed, not by it naturally happening. It's another benefit of organics. Shelf life potential of seeds can be detrimental based on the seed coat thickness. There's 3 different types of seeds: micropyle, mesopyle, and macropyle, and they all have different lifespans. So that usually goes on the size of the seed to the size of the seed coat thickness. The smaller the seed, the smaller the coat of thickness will be, in general, that is. So that means their shelf life will be lower. This breaks down a bit better inside of the seed. Shows the endosperm, inside of the germplasm, cumulus, sclerotium, and the radicle. Then the bran has nuclear tissue, the seed coat, which is the tester, tube cells, cross cells, the hypodermis and epidermis layers are in there, ready to start and grow. And it's got the bracts and the hair of the brush, is this type. Drying, threshing, processing, cleaning, and grading can be decided based on the seed characteristics and prevent mechanical damage. Determining the size of the storage organs, like endosperm, cotyledons help in increasing food production. Because the cotyledons are the solar panels. So once they can get out, they can start and grab the radiation from the sun, solar radiation, and convert that into energy and continue the growth. A bit more stuff on there's the pericarp on the outside, just under the outside, then the endoplasm, the cotyledons inside, coleoptile, and the pumilule radical. This is a few different types of seeds. This is the breakdown of this particular type, because there's monocots and dicots, and inside of this monocots you'll see there's only a single cotyledon, like rice, like what's some old plants? Gee whiz, there's heap ferns, those types, but you don't usually get fern seeds, do you? But that's an example of a monocot. Dicots have two. There's some more bits inside of the tester and the tagman and castor seeds. See, so the caruncle, that bit, that's the lumpy bit that you'll see on the end of the seed, the caruncle. It's pretty cool. And it's got all its stripes on the outside, seed coat, inside again, the tester. Tagman, just on the inside there, cotyledons, the embryo, and the caruncle up the top again. Tester and tagman are fused in green gram, black gram, and red gram seeds. See, this is an example of the gram seed and how they're both fused on the outside there. Okay. Soybean, French bean, and peas are a bit different. I've got the tester there, as it should be on all seeds, and the plumula, hypercotyl, and radicular. Oh wow, okay. And they're all, those three things are the embryonic axis. You see how big the storage cotyledons is? Massive. Well-developed endosperm. Some castor again. So you can see inside of it, it's got this radicle, very small, and the hypercotyl is very small, like epicotyl. And the cotyledons are a little bit bigger. Stacks of endosperm around the outside. So it's got to be broken down. And then the seed coat. Seed maturation. It's interesting to see the duration of different species of, or probably even genuses of, varieties, because they take different times. Some take, like I've noticed, paspalum grass develops right inside of the tissue first, so when it spits it out, you only see it for probably 10 days and then it's mature. What's another long one is buckwheat, where it takes 60 days, 2 months to mature. So it just sits there and keeps going. You think, look at this stuff, it's just going forever. And it slowly darkens and darkens and darkens. But if you don't get the seed mature, you're not going to get the right outcome when you want to try and plant the seeds. So it's really, really important to know the species or genus that you're working with, so you can get if you're breeding, you want the seeds to be mature. And not just branch mature, but the whole plant mature. So a good way to really tell that is nearly just let it die, because it goes through different phases, the seeds. I wonder if this will come up. Yeah, just let it go. The longer the better. You'll see, because in nature, once the plant goes to die, these will naturally dry up and start to open and drop them out. That's when they're ready, they've full mature, they're ready to go. So if you get it too much early, some things might be missing, and it's just like breeding. If you get a premature baby, you've got to try and keep that alive and going somehow to try and go through its processes that it needs. So seed maturation, a process comprising a series of morphological, physical, and psychological and biochemical changes that occur from ovule fertilization to the time when seeds become physiologically independent of the mother plant. Here we go. There's no timeline. Well, because that depends on the species that you're working with. So after the fertilization's taken part, that's when the male bits have fertilized the female bits and the seed starts to grow. Here's the timeline down the bottom on the x-axis. So this solid line is fresh weight, dot's moisture weight, and this one down the bottom here is dry weight. So you can see the start, fresh weight starts high, falls, dry weight starts after a while because the seed's got to grow. Then it starts to grow, and it's reached its maximum dry weight. And the moisture content, as you want, because you don't want the seed to be, as it's growing here, and its dry weight to be high, otherwise it's prone to fungal disease, some problems, if the moisture is going to remain high. So that's why it naturally goes lower. And this is in perfect scenario. And if you're not in, if you're in very moisture-rich environment, use biopesticides, and they will take care of it. But I'm not talking about that. This is to talk about seeds. So I suppose that describes, it goes through millet seed. I should stop, see if there's any questions. Let's see what we will be doing as well. Infinite Wonders. With the other live stream, I don't mean, LOL, don't mean it should be short if needed. And I'll probably go for about 20 or 30 minutes. This is probably going to end 20 or 30 minutes. Renee says, okay, well, good luck on your exam. Thank you, I'll need it, that's for sure. Hey, it's Mace. G'day, Mace. Nice to see you, buddy. And genetic memory files. Nice to see you, mate. It says, good evening, good peeps. I hope you all are safe and happy. Yeah, I got my crop breeding exam in a few hours, and I'm doing another live stream about half an hour's time on a study session for it. Back to this. Good luck, Aussie, rooting for you. Thank you for your nice words. GMF, Renee, and everybody. It's going to be hard, as you'll see if you watch the live stream, the next one, you'll see the exam, some of the questions I go through, the review questions, and they're blooming hard. Just, anyway, I'm talking about seeds at the moment. Get through this bit. Millet seeds is pretty much the same one that I went through before. Dicots again. So this is talking about the germination of the dicot. And well, it's got the epicotyl there, the hypocotyl, which is the stem that comes out, and the cotyledon leaves. As it starts to germinate, you see the white tip, which is a radical, and the hypocotyl starts to form, and the radical changes into the root. It starts to get its lateral roots. Ones are going sideways. And cotyledons form to get your first leaves. So that's why there's enough energy in that seed coat from the starchy bits around the outside to get it to cotyledons, to this stage here. So theoretically, if you get organic seeds, there should be no need to stimulate them with any nutrients to this stage. After this, it might be looking for something, but that's where your lateral roots will have already found their areas that they like, and they will have already put out their protons to ask to exchange for the elements that they require. Oh, here's the timeline, which is a bit better for seeds. Remember saying before it varies from 10 or 15 days, paspalum, that you might see on the outside, to 60 days in buckwheat. So it starts, by the time it germinates, fertilizes has taken place, and then it's got to start and grow. It's a process that happens. So up here, seed formation, differentiation, then mature, reverse acclimation, post-abscission, and it's ripening, optimum time to collect, and it starts to dispersal, post-harvest, aging and repair, and the risk of viability loss, or it slips into dormancy, that you've got to do dormancy techniques to break it out of it. The seed coat might go thicker, might be different elements that change inside of it, but you can always, there's cells inside of it, so worst case scenario, you can tissue culture. And you want to know what you're breeding with your seeds. Like this year I made, oh gosh, at least a dozen different varieties of seed. So I just know their times for when they're mature to get harvest at the right time. Because I don't want to pull them when they're not produced. I want the germination rate to be 98%. So if I put out 100 seeds, 98 I want them to germinate, which is happening. Seed development stages in peas. All right. So as it fertilized there, it's got the zygote down the bottom. The endocarp starts to form. The cells start to differentiate. You see it starts to form its actual structure in what it's going to form after it's developed. It goes through its first called the globular stage, where it looks like a globular. Then it goes through into its heart-shaped stage. And then, I can't remember the other stages. It's 4 stages it goes through. And then it's pretty much developed and it's ready for germination. Cotyledon leaves ready to expand, ready to bust out and photosynthesize. Permissible amount of moisture in seed at harvest. Well, 13% for rice, 12% for cereals, 10% for pulses and oil seeds, and 8% for vegetables. Why is that? Why don't they allow anything more? Well, it's because of the moisture stimulates the fungus growth, and when it's very moisturized, it's up above 90%, 92%, it stimulates bacteria growth, and that starts to produce enzymes and break it down. Use it as food. So you want to store it so it's going to remain in its state, so when you want to reuse it, it's fine again. And you have to dry it all out. Irrigation at the time of harvest will delay maturity, says this slide. Okay. Withdrawal of irrigation hastens maturity, leads to immature drying and loss of seed potential. Wow, there you go. Just as well, I'm into the organic style things, and I nearly wait till I see permanent wilting till I go and give water. So mine would be used to it. Rains at maturity will lead to fungal infection and insect damage, may quicken maturity. Oh yeah, because insects damage, so it may want to speed up, because when the plant is trying to reproduce itself, that's its end goal. So when it gets under stress and problems, it starts and reproduce itself. Hot weather, water constraints during maturity also lead to hard seed impulses. Here's another little cartoon of the pea germinating, going through a few of its phases. Events occurring, this is rad. If you're going to take anything out, this is one of the best slides. Events occurring in germinating seed. So what happens when a seed starts to germinate? You've got a seed sitting there and you want it to germinate. What's happening? It needs 3 things: the right temperature, oxygen, and water. Once water goes in, so it's got the first 2 things, oxygen, temperature. Water goes in, it gets imbibed through the bottom here. It starts producing gibberellic acid, which starts to put signals down through DNA, unlocks that. RNA signals produce a protein. which release the enzyme amylase, which starts to break down the starch. And that produces the sugar, which can produce the energy for it to start for its growth. There you go. It's pretty easily explained with good diagrams. So the requirements of water, requirements for seed germination, water, oxygen, and temperature. You can read them. The slide should have been before it. And once in the ground, there's two types of germination, epigeal germination and a hypergeal. So one, epi, mean outside, it germinates outside of the ground. Hypo means less than or under, so it germinates underneath and then starts going up. Here's some growth development stages down the bottom. The second day, 5th, 7th, 9th, and 11th, just showing its different stages and how the seed should go on. Quite a lot you'll look and you'll want to check it out at as they're coming up, because they'll get stuck. I'll get little seed coats stuck to them, or the tester on the outside will get stuck, and you just want to manipulate that off. I call it doing surgery. It's my garlic, I had a bit too, just last week. Two types of germination, I already said that. Hypo and epigeal. Okay. You can see this is one of the courses that I've done that wasn't from my normal provider. It's one done still through university, but they're all over the shop, as you can see. And the same with their teaching. So I try not to do courses from these people anymore. Epigeal germination. Should I shout them out? No, I don't think so. Phases of seed germination. Imbibes, where it takes in water. There's a latent phase where it starts to do its stuff. Then exponential growth phase. Dormancy. What's dormancy? It's a temporary suppression of visible growth of any plant organ containing an apical meristem or embryonic axis. What? That's just dormant. Temporary suspension growth of any plant organ containing. Okay, yeah, okay. That's all right. Plant dormancy. We are on to talk about seed dormancy. So that's the meristems, the halt, the ram and the SAM and the RAM. We're going to go into seed development. Oh, sorry. Quiescence. Seeds are not provided with adequate conditions like water, temperature, or air for germination. It's like seeds in storage. So it's quiescence. They just sit there. They can't do anything. This will go about seed storage too. You should always store your seeds in low temperature, low humidity. So once they're dry, they'll stay for extra amount of years if you do that. Dormancy. Seed dormancy. The seed remains ungerminable even through providing suitable conditions. So you have to snap it out of dormancy with a few different techniques. So the first bit is primary dormancy, and then the secondary dormancy. So primary dormancy contains physiological, morphological, morphophysiological, physical, and a combination of them all. Physiological dormancy in angiosperm plants caused by physiological inhibiting mechanism. So here's an Arabidopsis, tobacco, sunflower, that have gone in dormancy, and they've gone through their stages. You can see there, after 35 hours, it's not happening. 150 hours, that's nearly a week, it's not happening. It's dormant for some reason. So yeah, they could have, Next, 3 types of physiological dormancy: non-deep, intermediate, and deep. Physiological dormancy is gibberellic acid treatment. So non-deep, where seeds tend to germinate after a period of storage. There's one way of breaking dormancy. Get G3 gibberellic acid. If you're going to order some, gibberellic acid treatment, seeds will not respond but produce seedlings, because it's one of the 2 hormones that a seed requires to germinate, ethylene and gibberellic acid. Physiological dormancy caused by one or more water permeable layers of palisade cells on the outside. Combination dormancy, where seeds possess stimulants, physiological and both types. Methods of overcoming dormancy: scarification, which is rubbing them after ripening; stratification, by putting cold temperatures; use of dormancy breaking chemicals; light, leaching, alternate temperatures, maybe more warmer, and priming. What else can I add to that? Where's the chemicals? Well, maybe ethylene. Ethylene's major. Here you go. Scarification, injuring the seed coat to make it permeable for water. So somehow rub it down, get water into it. After ripening, soils may be stored to reduce the effects of dormancy. I was talking about low humidity, 3 Celsius, which is, what's that, 35 F? No, 40 F probably. Stratification, exposure of seeds to low or high temperature. Use of dormancy breaking chemicals like potassium nitrate, thiourea or gibberellic acid, or ethylene. Light will promote germination. Yes, it might promote germination, but once it germinates, take it out of the red light because it slows it down. Showing different examples there of the different shades of red that they've done it under to get the germination faster or slower. Leaching seeds in running water to wash off inhibitors. I'm right there. Seed is a special unit with mature ovule that consists of embryonic cells. It's going backwards. The caruncle, we'll discuss a bit on the top. Different phases, spoke about that. Spoke about that, germination. See how that's all over the shop? Exponential growth. It's a summary of it all. Different phases of certain morphological dormancy. Morphological dormancy. Cotyledons and hypercotyledon radicle are small in size. Morphological dormancy in celery, also smaller in size. Physical dormancy. Lettuce seed dormancy. Secondary dormancy induced by unfavorable environmental conditions. So it's when you don't have the right temperature or oxygen or water, I suppose. Photodormancy induced by light. Photodormancy. occurs by exposure to an excessive light. That's the last one. Just didn't see if there are any questions for a while. Good luck, says, yeah. Good luck, thank you for that, mate. The one who says, didn't know about gibberellic acid being used to help seed germinate. That's cool. Didn't know about a lot of stuff, but the gibberellic acid trick will be something to look into. Yeah, if you want to do that, well, if you, more so, it's ethylene. Didn't mention anything about ethylene. This is a great trick, though. You get an apple, look up the most volatile fruit for emitting ethylene. My voice is sore. Shit. And it's ethylene. Here's the gas you want. If you get, so you get an apple and leave it in with your seeds. The seeds will be put on the top of the water. You just leave it in there in a bag, and it'll induce it and help it. And that's one of the best tools that I use. An apple. Clean the apple, disinfect it, because it's going to be in tissue culture conditions. But yeah, I've done it without apple, and with it, big difference. And if you see it, it gets a triple response, it's called, ethylene triple response, where it gets a J hook of the root, it's short, and the hypocotyl thick. So if you put too much into the manual, that's the trick you can get to tell the difference. Why didn't it do that again? I want the big screen. Spewing. Anyway, it doesn't matter. 51. This has been a good first week. So if there's no questions, put them now, please, if you got anything to say, because I'm going to end the stream, because I've got to do another stream in 8 minutes, and my voice is sore. Oh, these are my things that I put on my arm because I've got sore arms. Oh. All right, people. Thanks for joining week 1. We'll do week 2 next week. Happy breeding, happy growing, good health to you all. Bye-bye. I've got a study session happening in 8 minutes' time. If you want to hang around, I'll be starting it again. See ya. Thanks for watching, everyone. Bye-bye.